students, this is our first lecture, Basics of Molecular Biology. In this subject, we are going to study cell at molecular level. Now, whole organism, as we know, is made up of different cells which unite together to form a tissue. All the tissues, different types of tissues, they combine together to form an organ, and whole organs together with the cells, tissues, blood vessels and etc. form whole organism. So basic structure of cell is composed of four biomolecules. These are proteins, lipids, carbohydrates and nucleic acids. So, in molecular biology, we'll particularly focus on nucleic acids, that is DNA and RNA, how they are synthesized, how they unite together, how they work hand in hand for signaling, proper signaling, how they form a protein and how these proteins work as building blocks. Now next is gene. Gene is unit of heredity. Now you know, into the cell DNA is complexed with the proteins and together it forms a chromosome. So the DNA segments that carries genetic information are called genes. Now it is normally the stretch of the DNA that codes for a type of protein or for RNA that has function in an organism. So gene is the segment of DNA which codes for a functional unit. This functional unit is either RNA or protein. Okay. These genes hold the information to build and maintain an organism's cells and pass this information to successive generations. That is, from parents to the child, this genetic information is passed from genes. As you know, when two sexes unite, that is the nucleus of sperm and ovum, when they unite together, there is recombination, that is the chromosome from male and from the female parent, they exchange their genetic material, most probably the genes, and they form a zygote which have few new characters and few characters which are similar to their parents. So these genes are the unit of heredity which are transferred from one generation to the other. Okay? What is genome? Genome is entirely of an organism's hereditary information. It is encoded either in the DNA or from many types of viruses in the form of RNA. In 99% of the organisms, DNA serves as genetic material. Only the viruses are exceptions, where in few viruses you will find RNA is present as genetic material. The genome includes both the genes and non-coding sequences of the DNA. Now the coding sequences, that is genes or exons, they code for a functional unit, that, and non-coding sequences codes for nothing, that is known as the junk DNA, which codes for nothing. They are known as introns. You will be surprised to know that out of the whole genome of organism or say human, only 2% genome codes for either RNA or protein. Rest 98 to 99% of the genome is non-coding, does not code for any of the protein or RNA. This is the comparative genome sizes of organism. Now see, humans size of the genome is 3.2 billion base pairs. That is 3.2 into 10 raised to 9 base pairs. Gene number present is 25,000 genes approximately. This is the density of the gene, that is out of say 10 raised to 5 bases or 1 lakh bases, only one gene is present. See, human genome size is very large as compared to the number of genes present on it. Size is larger, but number of genes present is very less. Only one gene is present per 100,000 bases. Number of chromosomes is 46. That is 23 pairs of the chromosomes are present. Mouse, mus musculus, 2.6 billion base pair DNA. Number of genes is almost similar to the human beings. Gene density is also similar to the human beings. That's why it is said that mouse genome is related, is very close to the human genome. Almost 99% similarity is there in between mouse genome and human genome. Chromosome number in mouse is 40. Drosophila melanogaster, that is fruit fly. The genome size is 137 million. Million is 10 raised to 6. That is 137 base pair into 10 raised to 6. Gene number is 13,000. Gene density, now see here, this genome is approximately 1000 times smaller than the mouse or human genome. If you see the Drosophila size, it is very small organism as compared to the human, and genome is 1000 times smaller than that of the human or mouse. But the gene density, if you see, one gene is present per 9,000 bases. Okay, gene density is higher than the humans and the mouse. Chromosome number is only 8. Then the plant, model plant Arabidopsis thaliana. Size of the genome is 100 million, that is 100 into 10 raised to 6 base pair. Number of genes 25. Average gene density is one gene per 4,000 bases. Means after each 4,000 bases one gene is present and chromosome number is 10. Likewise, here are the examples given for other organisms, for yeast, for E. coli and for Haemophilus influenzae. This Escherichia coli and Haemophilus influenzae. These are the bacterial cells. Okay. Yeast is single-celled eukaryote. This is Caenorhabditis elegans, which is again model organism. Okay. Here we will take example of Escherichia coli because this is model organism. Size of the genome is 4.6 into 10 raised to 6 base pair. Genes present 3,200 genes present, and gene density is one gene per 1400 base pair. Chromosome is single chromosome. Then why to analyze genome? This is for the prediction of the genes in uncharacterized genomic sequences. To obtain complete sequences of many genomes,